Question number 126. Diurnal acid cycle is first option as efficient as C4 cycle, second responsible for fast growth of succulents, third related to chloroplast dimorphism, fourth found in plants with scotoactive stomata. Diurnal acid cycle is CAM cycle, crassulacean acid metabolism cycle, which is seen in succulent plants. So this CAM cycle is as efficient as C4 cycle? No, it is less efficient as compared to the C4 cycle. Why? Because the CO2 fixation takes place during the night, and hence the amount of CO2 which is already present inside the cells can be only utilized to fix the CO2. Second, responsible for fast growth of succulents. Succulents show slower growth, so this option is also ruled out. Related to chloroplast dimorphism, there is no chloroplast dimorphism in the CAM plant. There is a temporal difference in the fixation of CO2. Temporal difference is the difference in the time of CO2 fixation. Fourth, found in plants with scotoactive stomata. Scotoactive stomata are the stomata which open during the night and remain closed during the day. So CAM plants have scotoactive stomata. So this option is correct. So the correct option for the given question is option number 4. Let's move on to the next question. Question number 127. Photorespiration is useful for plants as it, first option, protects plants from photooxidative damage. Second, provides net gain of 2 ATP. Third, enables Rubisco to show its oxygenase activity. Fourth, increases rate of CO2 fixation. Photorespiration is useful for plants as it provides net gain of 2 ATP. No, there is no synthesis of ATP during photorespiration, so it does not provide any gain of ATP. Third, enables Rubisco to show its oxygenase activity. Yes, photorespiration enables Rubisco to show its oxygenase activity, but the oxygenase activity of Rubisco is not useful for plants, hence this option is ruled out. Fourth, photorespiration increases rate of CO2 fixation. No, instead it leads to loss of the fixed CO2 in plants. So this option is also ruled out. So how it is useful? As it protects the plants from photooxidative damage. What is photooxidative damage? When there is this highlight intensity forms the reactive oxygen species if oxygen is also present in the high amount. So during the photorespiration, high concentration of oxygen is combined with the RUBP, hence there will be no photooxidative damage. So the correct option for the given question is option number 1. Let's move on to the next question. Question number 128. In C2 cycle, oxygenation and decarboxylation occur respectively in, first option, chloroplast and peroxisomes, second, chloroplast and mitochondria, third, mitochondria and chloroplast, fourth, mitochondria and peroxisome. First of all, you should know what is C2 cycle. C2 cycle is the another name for photorespiration or the photorespiratory cycle. In photorespiration, there are 3 cell organelles which are involved. First is chloroplast, second peroxisome, and the third is mitochondria. I'm just drawing it just to make you understand. So in chloroplast, when O2 combines with RUBP, and in mitochondria there is a release of CO2 which is called decarboxylation. So this is oxygenation and this is decarboxylation. So oxygenation takes place in the chloroplast and the decarboxylation in the mitochondria. So the correct option for the given question is option number 2. Let's move on to the next question. Question number 129. Select the correct match for C4 cycle. First option, first product of CO2 fixation, 3-carbon compound. Second, PEP synthetase, cold sensitive. Third, transportation of C4 acid, OAA, mesophyll cells. Fourth, regeneration of primary CO2 acceptor bundle sheath cells. This question is all about the C4 cycle. In C4 cycle, first product of CO2 fixation is a 3-carbon compound. No, why it is called C4 cycle? Because the first product of CO2 fixation is a 4-carbon compound, and that is oxaloacetic acid. So this is not correctly matched. Second, PEP synthetase is cold sensitive. Yes, PEP synthetase is required to regenerate the PEP, and this enzyme is cold sensitive. Third, transportation of C4 acid takes place in mesophyll cells. No, transportation of C4 acid takes place in the bundle sheath cells. C4 acid is transported to the bundle sheath cells. So this is mismatched. Fourth one, regeneration of primary CO2 acceptor in the bundle sheath cells. No, this takes place in the mesophyll cells. So this is also mismatched option. So the correct option for the given question is option number 2. Let's move on to the next question. Question number 130. Choose the odd one with respect to the 4 carbon acids formed during C4 cycle. First option, oxaloacetic acid. Second, malic acid. Third, aspartic acid. Fourth, pyruvic acid. So the 4 carbon acids are OAA. Yes. Aspartic acid, yes. Malic acid, yes. Pyruvic acid is a 3-carbon acid. It's an intermediate during the glucose breakdown while glycolysis. So the correct option for the given question is option number 4.